Right now, while you are hearing these words, a sheet of paper from 1976 sits inside a NASA archive box, and on it is a wavy line that a team of scientists spent the rest of their careers arguing about. It is not a photograph, it is not a rock sample, it is a graph, drawn by a machine on the surface of another planet, showing gas rising out of a spoonful of soil the way gas rises when something inside that soil is alive. Before the argument began, before the doubts, before it consumed one scientist's whole career, that line went up. This is the story of two landers named Viking, and the one question they were built to ask. 50 years ago this September, the United States put two machines on Mars to ask that question directly. One instrument said something close to yes, two others said no. NASA called the whole result inconclusive, filed it, and never asked that way again. This is what each instrument actually found, why the file never really closed, and why one of the scientists who built it spent 45 years refusing to let it stay shut. None of this was inevitable. 11 years before Viking landed, the first American spacecraft to reach Mars, Mariner 4 flew past the planet in the middle of July 1965 and sent back a small handful of photographs that quietly ended one era of imagination and started another. For more than half a century, ever since the astronomer Percival Lowell built an observatory in Flagstaff, Arizona specifically to study what he was certain were artificial canals crisscrossing the Martian surface, astronomers and a great many members of the public had wondered whether Mars carried canals, forests, even cities. Mariner 4's pictures showed something closer to the moon, a cratered, wind-scoured, apparently dead world, cold enough to freeze carbon dioxide and thin enough that liquid water could not survive on the open surface for more than a few minutes. If Mars still held life after those photographs, the thinking went, it would have to be small, hidden, and stubborn, not the kind of life anyone could see from orbit. That is the world Viking was actually built to interrogate, a planet that looked dead from a distance and that NASA had decided to check up close directly with instruments instead of guesses. By the middle of the 1970s, the United States had already put men on the moon and turned around to do it again, more than once. Mars was the harder target, and NASA decided to go after it with something more ambitious than a photograph. Two identical spacecraft, Viking 1 and Viking 2, each carried a lander built to do something no machine had ever done before, run a working biology laboratory on the surface of another world. The lander would scoop up Martian soil, feed it, warm it, and watch closely to see whether anything in it behaved the way something alive behaves. The whole biology package weighed about 15.5 kilograms, not much heavier than a large bag of dog food, and it had to survive a landing, freezing Martian nights, and months of continuous use without a single visit from a repair technician. Every part of it had to work correctly the first time, because there would never be a second chance to fix it by hand, tens of millions of miles from the nearest toolbox. Viking 1 touched down on the 20th of July, 1976, on a flat, rock-strewn plain called Crusade, chosen because engineers believed it was the safest place on the planet to put a lander down. Its descent engines used 18 small nozzles instead of one large one, spreading the exhaust out so it would not scorch or contaminate the very soil the biology instruments were about to test. Within 25 seconds of landing, the lander sent back its first image, a grainy black and white photograph of its own footpad resting on Martian gravel. If you had been standing beside it in that first quiet minute, the only sound you would have heard is wind, because Mars barely holds onto a noise. It sat there, unattended, farther from help than anything ever built by human hands, and it got straight to work. The landing came during the country's bicentennial summer, 6 weeks after the 4th of July, and newspapers that had spent the year looking backward at 200 years of American history suddenly had a reason to look forward instead. Television networks interrupted regular programming for the descent. NASA had originally hoped to land on the 4th of July itself, a symbolic pairing engineers eventually abandoned once radar images of the planned site turned out to be too rocky to risk it. By the time Viking 1 was safely down and the biology experiments were running, a country that had just watched the Apollo program end wanted a clean, historic answer as badly as any scientist in the room did. What it got, months later, was a single word, inconclusive, which is a difficult thing to put on a magazine cover. Inside that lander were 3 separate biology experiments, each designed by a different team, each built on a different theory of what alien life might actually do if you gave it the chance. The whole scientific program answered to Gerald Soffin, NASA's project scientist for the mission, who coordinated more than 70 scientists across the country and had to make sense of 3 teams that did not fully agree on what a positive result would even look like. The first experiment, called gas exchange, fed the soil a rich broth of nutrients and watched the surrounding air for changes, on the theory that living organisms would consume or release gas as they fed. If you have ever watched baking soda and vinegar fizz in a school science project, you already understand the basic logic. The second, called pyrolytic release, was led by the Caltech biologist Norman Horowitz, and it gave the soil a simulated Martian atmosphere laced with radioactive carbon, on the theory that anything alive would try to pull that carbon into itself the way plants on Earth do. The third was the labeled release experiment, designed by an engineer named Gilbert Levine, working with his research partner, Patricia Strott. Their instrument moistened the soil with a nutrient solution tagged with radioactive carbon, then simply watched for gas to bubble up, on the theory that if something in the soil was eating, it would also exhale. Picture the procedure the way Levine and Strott built it, because it is simple enough to hold in your head. You take a small sample of Martian soil. You add a few drops of nutrient solution, tagged so any gas it produces can be tracked. You seal the chamber, you wait. On Earth, in every soil sample tested this way before, a result like that meant bacteria were present and feeding. On Mars, within hours of the nutrient touching the soil, the detector needle began to climb fast, higher than several of the positive controls Levin had run back in his own lab in Maryland. It looked, on paper, exactly like life. Strott would later say plainly that their instrument had produced what she called a definite positive response, and that a great many people afterward found a great many reasons to argue that it was not. Here is the detail almost nobody outside the mission team knew about at the time. The signal was so strong and came in so fast that some members of the biology team reportedly began drafting language for how NASA might announce that life had been detected on another planet. That draft was quietly shelved within days once the first doubts about soil chemistry started moving through the team, and it was never revisited. What happened instead took years to unravel, and in some ways it still has not finished unraveling. All three biology experiments shared the same basic safeguard: a heat-sterilized control. A separate portion of each soil sample was cooked to a temperature high enough to kill any Earth-type microorganism before the same nutrients were added, on the reasoning that a properly dead, sterilized sample should behave differently from a live one if biology was really involved. Gas exchange detected a modest release of oxygen the moment the soil first met humidity, more than ordinary chemistry alone seemed to explain, but the reaction did not grow or repeat the way a living culture should, and most of it was eventually attributed to reactive minerals in the soil rather than biology. Pyrolytic release returned a weak but real positive signal across several runs. Carbon was being pulled out of the simulated atmosphere and into the soil, and that signal dropped sharply once the soil was heat sterilized, exactly what biology should do and exactly what certain non-biological catalysts would do as well. Of the three, only the labeled release experiment produced a large, fast, repeatable positive signal, and only the labeled release experiment lost that signal almost completely under full sterilization, then lost it again, less completely, under a gentler heating meant to slow biology without necessarily destroying every possible chemical explanation. Strong response while active, reduced response under mild heat, no response after full sterilization, is exactly the pattern a biology textbook would predict. It is also, Levin's critics pointed out at the time, roughly the pattern a heat-sensitive chemical oxidant already sitting in the Martian soil would produce with no life involved at all. 6 weeks later, on the 3rd of September, 1976, the second lander, Viking 2, came down thousands of miles away at Utopia Planitia, a broad plain in Mars's northern hemisphere, chosen specifically because it was geologically different from the first site. NASA wanted to know whether whatever the first lander found was a local fluke or something true of the whole planet. Viking 2 carried an identical instrument package, run by an identical procedure, on soil scooped from ground no machine had ever touched. The labeled release experiment ran again, and it produced, again, a rising curve that looked like metabolism. Two landers, on opposite sides of a planet, had each returned an answer pointing the same direction. Stand on that ground today, even just in your imagination, and you would see almost nothing to mark it as significant. No monument, no sign, nothing to tell you that this flat, wind-scoured patch of Utopia Planitia was the second place a machine from Earth ever asked the same question of Mars and got, again, a strange answer back. But a single instrument does not get to settle a question this large by itself, and Viking carried a fourth piece of equipment built specifically to check the other three. It was called the Gas Chromatograph Mass Spectrometer, and its job was narrow and unglamorous. It heated a soil sample until it broke down, then sorted whatever came off it to identify individual molecules. If Mars had ever hosted life, dead or alive, this instrument was sensitive enough to find the leftover organic molecules, the basic carbon-based building blocks that every known living thing on Earth is made from, whether or not anything in the soil was currently breathing. On both landers, at both sites, the Gas Chromatograph Mass Spectrometer found nothing, not a trace of an organic molecule, not from a fossil, not from a contaminant, not from anything at all. That single result did more to shape the outcome of the mission than the other two experiments combined. The reasoning seemed airtight at the time. Every living thing anyone had ever studied leaves behind organic chemistry, whether it is currently alive or has been dead for 1 billion years. If the soil truly contained microorganisms metabolizing nutrients, that same soil should also contain the organic residue of those microorganisms and their ancestors. It did not. Weighing a strong metabolic signal against a flat organic chemistry result of 0, the wider scientific community concluded that the labeled release result had to be some kind of exotic soil chemistry imitating biology, not biology itself. NASA's official position hardened into a single word: inconclusive, and in practice, that word behaved exactly like a no. Chemists offered a specific mechanism for how soil alone might fake the result. Mars has essentially no ozone layer, so ultraviolet light reaches its surface at intensities Earth's own chemistry never has to withstand, and that light can manufacture highly reactive, short-lived oxidizing compounds in surface dust, chemical cousins of hydrogen peroxide. Wet that dust with the nutrient solution, the theory went, and those oxidants would react immediately, producing a burst of gas that looked exactly like metabolism without a single living cell anywhere nearby. Nobody had actually detected these specific compounds in Martian soil in 1976. It was, at the time, an educated guess built to explain a result nobody wanted to call impossible, and nobody could yet call proven either. Gilbert Levine did not accept it. He had designed the experiment, run the positive controls himself, and watched the same curve rise twice on 2 landers at 2 sites, thousands of miles apart. And he could not make himself believe that was coincidence or chemistry alone. In the years after Viking, he built a company called Biospherics around related detection technology, later using some of that same underlying science to patent an inexpensive method for producing a sugar substitute called tagatose. And he continued to argue his case in scientific papers, even as the consensus against him hardened further. In 1997, more than 2 decades after the landings, he formally published his conclusion that the Viking landers had, in his professional judgment, detected living microorganisms on Mars. Almost nobody in the field agreed with him publicly. He kept publishing anyway. In the spring of 2012, on the 12th of April, Levin took the argument further. Working with Patricia Strott and a researcher named Joseph Miller, he applied a mathematical method called complexity analysis, a tool normally used to tell a living process apart from a non-living one by how much internal structure and unpredictability it contains, to the original 1976 data. The team reported that the pattern in the labeled release results looked statistically more like a biological process than a chemical one. It was not new data. It was the same nearly 40-year-old curve, examined with mathematics that had not existed when the curve was first drawn. The finding reopened an argument that a great many scientists had considered settled for a generation. None of this proved anything on its own. A curve, studied more carefully, is still just a curve. But it kept the question alive in a way one determined scientist working alone could never have managed by himself. While the biology argument simmered, the rest of the Viking mission quietly became one of the great successes in the history of planetary exploration, almost as a footnote to its own controversy. Each lander had an orbiter flying overhead, mapping practically the entire planet in a level of detail nobody had ever had before, and those orbiters found dry, branching channels that looked exactly like ancient riverbeds and catastrophic flood scars, strong evidence that liquid water had once moved across a surface now bone-dry. They photographed Olympus Mons, a volcano roughly 3 times the height of Everest and wide enough to cover a small country, the largest known volcano anywhere in the solar system. And the landers themselves refused to stop working. Both were designed for a primary mission of 90 days. Viking 2 kept transmitting from Utopia Planitia for about 3.5 years. Viking 1 kept going even longer, more than 6 years past its planned lifetime, finally falling silent in 1982 after a command sent from Earth accidentally overwrote part of its own memory. The biology question never got a clean answer. The hardware, unglamorously, outperformed almost every expectation anyone had for it. Then, in 2008, a different Mars lander, built for an entirely different purpose, found something that changed the whole conversation without meaning to. Phoenix touched down near the Martian North Pole to study ice and climate, nowhere near either Viking site, looking for water, not for life. Its chemistry instruments detected a salt called perchlorate, laced through the Martian soil in small but consistent quantities, present not only at its own landing site, but later analysis suggested more or less everywhere across the planet. Nobody on the Phoenix team was thinking about Gilbert Levin. Perchlorate interested them because it lowers the freezing point of water and might explain how liquid brine could survive on a planet this cold. Almost as an afterthought, a few scientists went back and asked a very different question. What would that same perchlorate do inside the heated oven of the Viking gas chromatograph mass spectrometer? The answer, published a couple of years later, was the loop this story had been waiting to close. Heat perchlorate to the temperature the Viking instrument used, and it does not sit quietly. It reacts with any organic material present and destroys it, producing, as a byproduct, two specific chlorine compounds. And those two compounds, chloromethane and dichloromethane, were exactly the same two chemicals the Viking gas chromatograph mass spectrometer had detected on both landers, at both sites, back in 1976. At the time, the mission team had waved that detection away as terrestrial cleaning fluid, contamination carried along from Earth on the spacecraft itself. It was very likely never terrestrial contamination at all. It was, more probably, the ash of Mars's own organic chemistry, destroyed by the very instrument built to find it at the exact moment it was being searched for. Think about what that does to the original contradiction. The gas chromatograph mass spectrometer's null result had been treated for more than 3 decades as the single fact that outweighed everything else, the flat no standing against the labeled release experiment's yes. If the soil's own chemistry was quietly incinerating the evidence before the instrument could properly read it, then that null result was never as clean as it looked. It was not solid proof of absence. It was, at best, proof that the method of searching may have destroyed the very thing it was searching for. The picture got more interesting again in 2018, when a completely different rover found the very thing Viking's instrument had been built to detect, and never did. Curiosity, using a far more sensitive instrument called sample analysis at Mars, drilled into rock more than 3 billion years old inside Gale Crater and found organic molecules preserved inside it, simple compounds related to benzene and propane. Nobody claimed those molecules were evidence of life. They could have arrived on meteorites, formed through ordinary geology, or simply survived from an era before Mars lost most of its atmosphere, with no biology involved anywhere in the chain. But the discovery settled something Viking's null result had always left open. Organic chemistry can survive in Martian rock, in the right conditions, protected from the surface radiation, and reactive soil chemistry that appears to scrub it away everywhere Viking happened to dig. Levin lived to see that reversal, and he treated it as vindication, though he never lived to see the argument formally settled, because it still has not been. He kept publishing into his 90s and kept giving interviews, and in a piece for Scientific American. In 2019, he put it as plainly as he ever had, saying he was convinced they had found evidence of life on Mars back in the 1970s, and that history would eventually agree with him. He died in Bethesda, Maryland, on the 26th of July, 2021, at the age of 97 from a tear in his aorta, the body's largest blood vessel. He died 6 days after the 45th anniversary of the Viking 1 landing that had shaped the rest of his working life, close enough to that anniversary that it is hard not to notice. If you have ever refused to let go of an argument you knew in your bones you were right about, even after everyone around you moved on, you have some sense of what those 45 years must have felt like from the inside. Say plainly what is actually true here, because this channel does not exist to manufacture a verdict where none exists. The scientific consensus, then and now, is that Viking did not detect life on Mars. Perchlorate chemistry, ultraviolet radiation breaking down organic molecules at the surface, and other non-biological explanations for the labeled release curve remain, to the people who study this professionally, the more probable reading of the data. Norman Horowitz, who ran the pyrolytic release experiment and spent his own career convinced that carbon-based chemistry made life elsewhere plausible in principle, concluded from his own instrument's results that the Martian surface, at least in the too-thin patches of it Viking sampled, was almost certainly lifeless. That is the mainstream position held by careful, serious scientists, and it deserves to be reported honestly as the mainstream position. But Gilbert Levin's dissent was never a fringe conspiracy. It came from the person who built the instrument, and it was defended with real data, real mathematics, and a genuine unresolved gap in how the negative control was interpreted. Uncertainty is not the same thing as an even split, and it would be dishonest to pretend both sides carry equal weight. It would be just as dishonest to pretend the losing side was foolish. This would not be the last time a Mars life claim divided the field this way. 2 decades later, in 1996, a NASA-led team announced possible fossil evidence of ancient microbial life inside a Martian meteorite found in Antarctica. And that claim, too, generated enormous excitement before slowly losing support as non-biological explanations for its odd structures accumulated. Extraordinary claims about life on Mars have a way of arriving with real data behind them and leaving years later still argued about rather than resolved. Here is the part that explains why this argument still has no ending. NASA has not flown another direct biology experiment to Mars since Viking, not once, in all the years since. Every lander and rover that followed, Pathfinder, the twin rovers, Spirit and Opportunity, Curiosity, Perseverance, was built around a more cautious strategy that scientists came to call follow the water, hunting for the conditions life would need rather than testing for life directly. Part of that caution is technical. A positive result from one automated instrument sitting on a planet many minutes away by radio with no way to run an additional test on the spot is exactly the kind of ambiguous data that took Viking's own team decades to argue about. Part of it is institutional memory. Viking cost the country a huge sum in today's money, promised a clean answer, and delivered a 50-year argument instead. The safer approach, the one NASA has chosen mission after mission since, is to gather rocks, characterize environments, and eventually bring pieces of Mars home to laboratories on Earth, where a result can be checked by any scientist with a microscope, not trusted from a single machine's readout, transmitted once from tens of millions of miles away. That return mission has faced years of its own delays, redesigns, and rising cost estimates, which is its own reminder of how difficult it still is to get a clean, trusted answer out of Mars, even when nobody's asking about life directly. If you were old enough in 1976 to remember any of this unfolding on the evening news, you already know what that kind of anticipation felt like. A country that had just landed on the moon, assuming the next great answer was only months away, then quietly learning that some questions do not resolve on schedule. That sheet of paper from 1976 is still sitting in an archive somewhere, and the line on it still climbs exactly the way it did the day it was drawn. Nothing about the data itself has changed since Gilbert Levine first watched that needle rise. What changed is everything around it. A discovery made on a completely different mission in a completely different part of the planet that quietly reopened a case almost everyone had agreed to close. That is the real shape of how Mars tends to answer us, not with a clean yes or a clean no, but with a result that takes another spacecraft, another decade, and another argument to even partly understand. If you want more real mission history told straight. Without the exaggeration, subscribe to this channel. Right now, on the surface of Mars, sample tubes sealed by the Perseverance rover are sitting in the dust, waiting for a mission that does not exist yet to come and carry them home. And when that finally happens, this argument may get closer to the kind of answer Gilbert Levine spent 45 years asking for. Thank you for watching.